for adaptive. I hope a lot of you guys have been working on the bonus, watching those videos and doing your drawings, right, and engaging with this material. I find that is definitely the most helpful, right, for you guys for learning this. And so this morning I wanted to start out with, I did this one. I wanted to go over this animation. So let me plug this in. Make sure I have my audio on.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or cell proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells.
Let's be more specific here, though. What type of T cell is going to kill an infected cell? What type of T cell? A cytotoxic T cell, which has CD8, right? Remember 8, eat, right? Destruction, think of that. Right? And of course, CD8 goes with MHC class I, which is found on all nucleated cells, right? So again, this is going to be an interaction with a CD8 cytotoxic T cell because CD8 and MHC class I is that special handshake, right? Those are the two cells that can communicate with each other. And therefore, it's going to lead to destruction of this cell. Okay? I can't stress enough. I know it sucks for memorization, but as I said, try and tell yourself little stories, whatever it is, right? Because this is a very important interaction here. Okay? Knowing that secret handshake, right, is important.
Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles, such as viruses and bacteria, by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
Now, who is this antigen-presenting cell going to communicate with it because it's expressing MHC class II?
Helper T cells?
Yes, helper T cells, right? What CD marker do helper T cells have?
CD4.
CD4. Remember, 4H, right? 4 for helper cells. Now, this could be a macrophage, or who else tends to do antigen presentation and activation of T cells? Dendritic cells, right? And who else will the helper T cell help out that will present antigen other than a macrophage or a dendritic cell? B cells, right? Which make antibodies, right? Their B cell receptor is an antibody. What do we call that cell, a B cell, when it's actually secreting antibodies?
Plasma cell?
Plasma cell, right? Plasma cell. So now we're going to look at virus protection. Do you guys remember what cytokine class was important for alerting neighboring cells when a cell was infected with a virus? Interferons, right? Interferons. So that's one of the protection methods, right? That's, having receptors inside the cells that detect viral nucleic acid, sending out that signal to neighboring cells. Those neighboring cells make enzymes to destroy the virus if it gets inside of them, right? And basically destroy themselves in the process, but we stop the replication cycle of the virus. That's one way we can protect against viruses, is interferon. Do we want only one way to protect ourselves against something? No, right? So we're going to talk about two other ways, right, that we can detect viral infection. Now, right now, we already looked at one of them that they're going to show again here, is that every cell is always presenting antigen on its surface via MHC class 1, right? So who's going to come along and destroy that cell if it recognizes that antigen that MHC class 1 is presenting? MHC class 1 goes with what? CD8, right? MHC class 1 goes with CD8. And what do we call CD8 T cells? Cytotoxic T cells. So what is it going to do to this virus-infected cell? It's going to destroy it. So this is basically what we've just watched a moment ago.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complex with class 1 MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class 1 MHC.
Yep. So here's MHC class 1, right? Here's the T cell receptor. There's the antigen. What is this? What is this molecule or molecules? This is a cytotoxic T cell. So this is CD8, right? It's the CD8 that allows them to communicate, remember, with MHC class 1. All right, secret handshake.
The cytotoxic T cell then releases cytotoxins, which induce apoptosis in the infected cell, and perforin, which causes perforations in the cell membrane. Self-proteins are also degraded, and the fragments presented on the surface via class 1 MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
So it said that if it was a self-antigen, which they didn't show you here, presented, a cytotoxic T cell is not going to recognize that and is not going to kill that cell because it's a self-antigen, right? It's our own proteins. Why is that? That we shouldn't be in the key word there, have any T cells that recognize us.
Does that kill us?
Yeah, that we would be attacking ourselves, and we certainly wouldn't want to do that. But why is it that we don't have T cells that recognize us? Do we make maybe T cells that could potentially recognize us? Yes, we do. Because remember, how do we make the T cell receptor and antibodies? We pick our lottery numbers, right? It's random. We randomly rearrange our DNA to make T cell receptors and antibodies, the B cell receptor. You guys remember? They picked the different parts. I don't have to play that animation again, right? And I did that additional drawing. I need to do a PowerPoint for that, right? So we randomly make these. So could we make a T cell that recognizes our own antigens? Yeah, it's definitely possible, and unfortunately it does happen for certain diseases as well. This is when we call it autoimmune disease, right? When the immune system attacks itself. But what system and where does it happen do we have in place to get rid of T cells and B cells that recognize us. So first of all, for a T cell, where does that happen in our body? Where do we eliminate the ones that recognize us? In the thymus, right? Again, T cells are named after the thymus. That's where they become helper T cells or cytotoxic T cells. But also, very importantly, right, that's where we get rid of or turn off the ones that recognize self. We don't want that happening. Make sense? Okay. So, natural killer cells. We haven't talked about these lymphocytes in a while. This is probably the last couple of slides in the PowerPoint. There you go. One of these outcomes we've already talked about. It's one of the antibody outcomes that involves natural killer cells. Does anybody remember that? It's an abbreviation commonly written ADCC. But what does that stand for? A is for what? No. It's a product of our adaptive immune system. What's the most common product we can easily measure? You know this. We're trying to measure them right now, right? Antibodies, right? Yes. Antibody dependent cellular cytotoxicity, because we're going to do what to cells that are infected with a virus? We're going to kill it. And the natural killer cell is the one that's going to kill it. But how does that natural killer cell know to kill that cell? That cell is coded in? No. Antibodies. Antibodies. Because it's antibody-dependent cellular death we're talking about here, right? So it's the antibody that is recognizing the viral proteins. Because what happens for a lot of the viruses that infect our cells is they're going to make their own proteins and insert them actually purposefully into our membrane to turn our membrane into their envelope. In that process, if we've got antibodies, maybe because we've been vaccinated or we've seen this virus before, our antibodies are going to bind to the surface of that virus-infected cell. When that happens, natural killer cells, when they come along, they can grab onto the Fc portion of that antibody and kill that cell. But there's only one class of antibody that can do this. What is it? IgG. 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 So that's one way we protect. And one of the most important things, or maybe unfortunate, about natural killer cells is, do they have a receptor on them that can recognize specific antigens? No. T cells do and B cells do. Natural killer cells do not have antigen-specific receptors on their surface. So they rely on other things in our immune system to help us with recognition. So for instance, antibodies, as it comes to antibody-dependent cellular cytotoxicity. Oh my God. Tongue twisting me today. So that's it right there, right? Antibody-dependent cellular cytotoxicity. The other protection that natural killer cells provide against viral attack is that some viruses, when they get inside of our cells, they shut down that system of the cell presenting via MHC class 1, right? Because your cells are always doing this, right? They're always putting their self-antigens on the surface. So that way, if a cytotoxic T cell comes along, right, he's going to check it out and be like, oh, I don't recognize any of this, you're fine, right? You're checked out, you can move on. Some viruses shut down MHC class 1 presentation. They stop the cell from putting MHC class 1 on the surface. So can a cytotoxic T cell recognize if it's infected? No, because remember, it needs that MHC class 1 holding the antigen so that its CD8 marker comes along, right? They do their special handshake. The next thing is, do I recognize this antigen? Well, if they can't even communicate because no MHC, then the virus can hide inside of our cells. Do you see that? So we need a protection mechanism that lets us know when this is happening. So notice no MHC, so natural killer cell comes along, right? And he's checking for MHC, class 1. Doesn't find it. Alarm goes off, right? That's not normal. This cell should be presenting MHC class 1. Why isn't it? What's going on inside that we need to know about? Something bad, right? When did your kid not give you their report card? When there's something bad on it, right? When it's all A's, they're like, Mommy, report card, right? My son forgot to give me his, but I knew what was on it already, right? And there was actually something really good. But I communicate with my son's teacher, so I already knew before the report even came out, right, what he was deficient in, what we need to work on. But think about that, right? If you've got something bad happening, right, inside, you want to avoid detection. You don't go around going, you know, here's my report card when you have an F, right? Because you know that's bad news. Does that make sense? So we have protection against this. Nowadays, you know, I just text his teacher. Right? So this is our scout. This is our extra protector. And as his name implies, what is he going to do when he runs across somebody who's hiding something? He's going to kill him or expose him. But in this case, right, if you're not doing what you're supposed to in the body, you are dangerous. You need to be eliminated. We're actually going to kill you. I'm not going to kill my kid over an F, though. Right? But so Bridget asked the questions, well, what happens when HIV virus is latent? And first of all, what does latent mean? No. It means it's hiding, right? So what happens is that the virus information is in there, but it's not replicating, right, in that cell. So this is not going to help us because that, I don't think, but don't quote me on it, I don't think when the virus is latent it stops MHC presentation. But also, when it comes to HIV, what's the major problem with this? What cells does it infect? Helper T cells. More specifically, helper T cells. And do you know how it gets into a helper T cell? Every virus, what's the first step in viral infection? Good review for our final. What's the first step? What does that virus have to do? It has to attach. Guess what molecule it attaches to on helper T cells? What makes it a helper T cell? CD4. Helper 4H. Don't forget 4H, right? 4H. CD4 is helper T cells. That marker that our cells are using to communicate with each other is actually what the HIV virus attaches to. And that's how it gains entry into helper T cells. And why it's called human immunodeficiency virus, because it can debilitate the T cell population so extensively that an individual's immune system becomes deficient. Because as we've just discussed, those helper T cells are really important, right? They're going to help out what cell? A lot. B cells, right? So if they can't help B cells, what can we not do? We can't make antibodies, right? And it's not that we can't make antibodies at all, right? Because do we need T cell help to make all antibodies?
No.
No. We need it to make anything other than IgE. What do we always make? And a B cell can do this on its own. What class of antibody can it make on its own? IgM. But remember, IgM's limited, right? It's the mega big pentamer, it gets stuck in the blood, it doesn't really go anywhere else. So we're not going to produce any IgA, which means what's not going to be protected with antibodies? We don't produce any IgA if we don't make that class switch from G to A. What parts of our body are going to be susceptible now to infection? So it's in your secretions, right? IgA is in your secretions. What secretions?
Tears.
Your tears, your saliva, your mucus. Your mucus is huge, right? Protects your whole respiratory, digestive tract, right? So what's the most common thing that gets people who progress to the state where their immune system is deficient because of HIV infection, that we say they have AIDS, which is acquired immunodeficiency syndrome, because at one time we didn't know what was causing these people's immune system to become deficient. So now their immune system is deficient. So they're not producing IgAs for one, right? Probably not producing any IgG either. What's the number one cause, right, of death in AIDS patients? Huh? Right, but more specific, anyone know what system of the body usually gets infected? No, your immune system is deficient, and it's in respiratory, so you're getting warmer, so your lungs, what's the most common infection in the lungs that will kill people? Pneumonia. And again, this all stems from the fact that you have limited protection because, one, you're not producing all these other antibodies, right? You're not producing IgA, so you breathe this stuff in, and you don't have that extra level of protection. Infection ensues, and you die. Helper T cells also help out. Other than the B cells do this whole class switching and making all these other classes of antibodies, who else do they help out that are really important killers in your body? Who's the big eaters? Macrophages, right? Helper T cells help out macrophages become more powerful killers. If you don't have any T cells because they've been destroyed by the HIV virus, can you do that? No. And the even worse news, right, is that your helper T cells, right, are infected. So what is your immune system also going to do? Your helper T cells are infected, right, with a virus. So one, they need to do their job for their immune system. But number two, since it's infected with a virus, what is your immune system going to do to its own helper T cells?
Destroy them?
Yeah, it's going to kill them. It's going to kill them, right? Either interferon is going to help kill them, right, when some cells tell other cells that the infection is there, or the cytotoxic T cells are going to kill them, or natural killer cells, right? If you have antibodies against the HIV virus, which, you know, again, the virus right away is not going to eliminate all your helper T cells. So you are going to mount some type of response. The problem is if you can't get that infection under control, right, you can't stop the replication cycle of that virus, you're losing helper T cells that you need for your response in the process. So this is why when someone has HIV, they monitor their helper T cell level, because that's the population of cells that's most affected by the virus. That makes sense to you guys?
So once the AIDS stage is reached,
Mm-hmm.
is the person still considered a host for HIV? Because it can spread HIV around since...
Yeah. At this point, we don't necessarily have a cure for HIV, right? Once infected, always infected. So the bad news for the individual when we say they have AIDS is that their immune system has dipped so low in protection, their T cell count is like basically nothing, their helper T cell count, they can't defend themselves against secondary disease, right? So they die from things like pneumonia. Are they still infectious? Absolutely. Because we haven't cured them.
Even though T cells, or the helper T cells.
Right, yeah. Well, because it doesn't mean they're all gone, right? And that's the main population, and that's why it causes the immune system to be deficient. But it can infect macrophages as well. Yeah. HIV virus can infect macrophages as well, which is another really bad news situation, right? It's one of our major defenders. I mean, we still got neutrophils and dendritic cells, but, I mean, macrophages are, really important resident cells for our defense. Make sense? Right. So there's a lucky population in Africa, apparently, that won the genetic lottery. And the reason why I say that is because, again, we make changes and change happens to our DNA over time. They have genetically different CD4s. Different enough that the HIV virus does not recognize their CD4. They're naturally immune.
Is it because they already been exposed or something?
No, I mean, like I said, so for instance, sickle cell disease is very prominent in Africa as well because they have malaria. And so there's this selective pressure because individuals that are heterozygous for that, where they have one good gene and one bad gene, not all their cells become sickle cell, but about half of them do. Those cells are immune to infection by the plasmodium, the protozoa that causes malaria. So it's beneficial to have one defective gene for sickle cell. The problem with that, though, is that if you're heterozygous and your mate is heterozygous, your children potentially could be,
Homozygous?
right? So Punnett square, right? So, here, grab the marker. So if you're heterozygous, and I won't use S because it's hard to see uppercase and lowercase, but typically what we would write, so let's do B's because B's are easy, right? Big B, little b. So if you're a heterozygous, you have one good gene, right? So we'll say that's a capital B, and usually the defective gene we do in lowercase would be little b. So if you and your spouse are that means both you and your spouse are going to give either big B or little b to your children. So when you fill in the square, you could potentially give two good genes, totally normal kid, no sickle cell at all. Or your children could be heterozygous, just like you, right? They got a little b from one parent, big B from the other parent, and so they're just like you. They partially have sickle cell, not enough that it's still full-blown sickle cell anemia, but enough that they have those sickle cells, so when they get infected with the plasmodium, it's not able to replicate. It's not able to get into enough red blood cells. It's not able to replicate. About half your kids, probability-wise, could be just like you. A fourth of them could be normal, which in the case of malaria is not good, right? Or, worse than malaria, they get both defective sickle cell genes. They don't make their hemoglobin properly in their cells sickle cell. All of their cells sickle cell. So now they have full-blown sickle cell anemia. So the problem is sickle cell anemia persists in that population because this is beneficial. But if you're heterozygous, then you have the possibility of 1 in 4 of a kid having full-blown sickle cell.
My teacher from high school, she had 3 kids, and her son doesn't have it at all. Her 2 girls had real bad. She used to have to leave school and everything. Her 2 girls used to stay in the hospital.
Yeah.
Her son is fine.
Yeah. Because he didn't get it.
The dad,
Yeah, dad has full-blown sickle cell anemia.
The mom didn't have it.
Right, and the mom's normal, right? So it's this Punnett square for that family, right? So mom's normal, right? She just gives, no, mom can't be normal then. If she has children with full-blown sickle cell anemia, she's got to be heterozygous. So because she's heterozygous, it seems like she's normal, right? Because she's only has partial sickle cells. And if the dad has full-blown sickle cell, he's just giving sickle cell genes, right? So the kids could be heterozygous like the mom, right? There's a 50-50 chance that the kid is heterozygous, or the kid has full-blown sickle cell. So the thing is that when you have these environmental pressures like malaria, right? Like I said, it's beneficial to be this. But with being heterozygous, you have the chance of having that disease, right? And these genes are going to persist in the population. Where if this wasn't beneficial, like people who are heterozygous don't survive malaria, then they wouldn't continue having children and we wouldn't have this disease anymore. Does that make sense to you guys? So in Africa, think about this. If you're not killed by HIV, then you grow up and you have kids, right? And the reason why they're not being killed by HIV, one of the reasons is they're naturally resistant. Their code is different. They make a different CD4, so therefore their kids get that, right? And then their kids are resistant to HIV, right? And you continue on this population. The rest of us, though, are jealous because they literally won the lottery, right? The genetic lottery. We can't steal this from them. We can't use it as therapy, right? This is their genetics, right? You can't steal their ticket, right? Not in this case.
Are those CD4 receptors still similar enough to activate the B cells?
Yeah, obviously. Otherwise, then they would have a deficient immune system and they would die from that. So the question Dan asked was, does their CD4 still work? And obviously it does, because otherwise they would have a deficient immune system and they would die from that. Yeah. So. Yeah. So of course, we asked the question. It's like, why are these people not getting this infection, right? And they were able to trace it back to the CD4 that they have is genetically different enough that the virus doesn't recognize it, doesn't attach to it, therefore can't cause infection in those cells. Does someone else have a question? Okay. So disease states too, it's a lot of times, again, it's what's not going right? Which brings us to our next lecture, which is on disorders. So if you can understand the process, right, you can understand what's not going correctly, right? And you guys did a really good job with that, and thank you for asking about HIV, right? Because that's a really good opportunity to say, okay, why is this bad, right? What's happening here, and why is that not good for us? So for immunological disorders, and for your current textbook, which I'm proud to report, we voted to get rid of. So come fall semester, they won't have to read Horrible Prescott like you guys did. They'll have a new book to read. So I'm excited for my future students. I'm sorry that you weren't the future students, but the good news is change will happen. So in the current book, disorders are lumped into this adaptive chapter, right? It's one big, huge monster of a chapter, right? To say the least. In other book that I use for the non-majors class, I do separate it out in a separate chapter referred to as disorders. So that's part of the reason why I have a separate lecture on it. But I also believe, too, that, sometimes breaking things up a little bit would help us out, right? But it is related to adaptive. And just like we were talking about HIV, it's important to understand how things work. And so then when things don't work, We understand what's going on, what's not working properly. So there are what we call hypersensitivities, right? So hyper means what? Increase, right? So we're more sensitive than we need to be against certain things, right? We refer to these as hypersensitivities. One of them, type 1, we commonly refer to as an allergic response, right? It is a response to something that will not actually harm us, but our immune system responds as if it is pathogenic, dangerous, bad.
But that's what allergies are?
Yeah, they're hypersensitivity. We're overly sensitive to stuff that won't actually hurt us. Pollen will not grow in your body, right? It just won't, right? But our immune system responds like it will, right? Like it's a bad guy that got in and we need to eliminate it. Aggressively at that. Autoimmune disease, as it implies, auto meaning self, immune, right? Our own immune system is attacking us. So this is a defect where we're not recognizing self as being self, right? We're seeing it as being something foreign. Immunodeficiency, right? We know that HIV, it's named after the fact that this virus, because it affects our helper T cells, can cause our immune system to be deficient. That's something we acquire, right? That's why it's called AIDS. It's acquired, right? You acquired this infection. Can you be born immunologically deficient? Yes, right? And again, this stems from your genetic predisposition. Do you have all the information to do the job that you're supposed to do? Some people are born deficient, where their immune system does not know how to do certain things. And there are different types and levels of deficiencies, right? Some are pretty mild, barely affects the individuals. Others are much more severe. So much so that most of the children are actually not even born alive. They die in utero from infection.
Miscarriage?
No, from infection. Which would potentially result in a miscarriage, yes. Not going to term with the pregnancy. If they do make it to birth, right, people laugh about this, right? It's not funny. But the bubble boy, right? Everyone's seen the movies and the talk about the bubble boy, right? A boy who had to live in a bubble. Why did he have to live in a bubble? We didn't want to expose him to pathogens so he wouldn't get infected. Why? What was wrong with him? His immune system was severely deficient. It's what's called SCIDs, severe combined immunodeficiency. And it happens. I mean, it's not a movie thing. It's not a made-up thing, right? It's a real thing that really happens. It's rare, but it happens. And so those individuals have to be extremely careful to the point where, you live in a bubble as to not be exposed to things that would make you sick. Otherwise, you don't have a functioning immune system. You will die. So there's two levels of it. You can acquire a deficiency, especially with HIV. We know that can cause your immune system to be deficient. Or you can be born genetically deficient. Is HIV the only way that you could acquire an immunodeficiency? No, right? So what do we need for our immune systems to function properly? What do we need for our body to function properly?
What?
Nutrition. What's next? What do they tell you to do if you want to be healthy? Let me put it that way. Drink water, eat food, exercise. What does exercise help with? Other than, heart and all that other good stuff, body-related stuff. increase, yeah, regulates hormones, right? And these types of things affect stress levels, right? And stress is one of the major things, right, that can really affect the body and really affect the immune system, right?
So is that why they do the cortisol level?
Yes. Yeah, cortisol is one of those stress hormones, right? It can be debilitating to your system, right? So when do you see people suffering from recurrent disease? Like every semester, right, around this time, I see people, at least one person on campus with a cold sore. Cold sore is a herpes virus, once infected, always infected. And it will reactivate, right, and multiply that virus during finals, right? Because what do people not do during finals that debilitates their immune system? They don't rest. They don't eat properly. They eat out of vending machines, right? And they are stressed, right? Bad news for your immune system. So people who live in countries, or even in this country, that don't have access to maybe food even, or proper food, right? They're stressed over that, right? Don't have proper housing. Can't get proper sleep. Are they more susceptible to disease? Yeah. Malnutrition, right? All these things can debilitate your immune system. So, and did any of you guys fill out that table? Right? I had posted an extra table just for disorders. Anybody actually do it? No, you just printed it out? All right. I'm trying to go to it. Here it is. Okay. So, yeah, this is it. I posted it separately in a separate PowerPoint all by itself. Not filled in. Okay. So, there are 4 different hypersensitivities that we're going to talk about, right? So, this is a good summary of it. And it's going to be the focus of our lecture for Tuesday, which will be our last lecture, y'all. That's it. Test on Thursday, right? On all of this stuff. Yeah. So, typically they're numbered. Type 1, 2, 3, and 4. But that doesn't really tell you a lot, and I certainly don't want you guys memorizing stuff that isn't going to really stay with you, right? So although I will, as I lecture, say type 1 and type 2 just for shortness, sometimes I will just refer to them by their numbers. But for testing purposes, right, and for your own purposes, you should learn the full name of these hypersensitivities. So for type 1, the full name would be immediate IgE Mediated. So in this name, in this title, this gives us a lot of information, right? Immediate, which means this reaction happens fast, right? Who's the main offender, you could say, for this hypersensitivity? It's a particular class of antibody. It's IgE, right? So that tells you a lot of information just in its name. The next one is cytotoxic. So cyto means cell. And toxic means death. So we're going to be talking about cells being killed with this hypersensitivity. And that's important, right? We're going to be talking about cells. The next one is immune complex mediated. Anyone know what an immune complex is? So what's a very common product of our immune system that I absolutely love? They're proteins, and they're just amazing. They can bind to a cell, and if they're IgG, they can cause antibody-dependent cellular cytotoxicity by natural killer cells. They're antibodies. What do antibodies bind to? Antigens. So guess what an immune complex is? Antigens and antibodies. So what are we going to talk about with this one? We're going to talk about Antigens and antibodies. That's 3. Immune complex mediated is type 3. And in the current textbook that you guys have, it doesn't go really well into type 3. So I'm going to post and give you guys a handout on Tuesday about type 3. I'm going to post the handout and I'm going to hand it out in class because it's that important. Delayed cell mediated. So again, tells you a lot in this, right? It's delayed. It doesn't happen immediately. It's cell mediated because as you guys will find out as we fill in this table right now, all the rest of these are dealing with antibodies. Even the cytotoxic one, it happens because of antibodies. This last one, as the name implies, is it antibodies? No, it says what? Cells. So it's going to be immune cells that are causing the destruction here, or the reaction. So for each one of these, there are different type of antigens that can cause this type of hypersensitivity. And I'm just going to open all of them up. Soluble means what? It dissolves in solution, right? So these are antigens, you know, that can get into our blood, into our body fluids, and be kind of soluble, right, in solution. For cytotoxic, remember, we're destroying cells. So where are these antigens? They're on the cell. What could you get from someone else? What common cells do we give to other people all the time? And we can run into problems because those cells have different antigens on the surface than you have. Red blood cells, right? So we discovered this hypersensitivity when we started doing blood transfusions, right? When we started giving blood to other people. And then there's one other problem women have to worry about, right, as it relates to blood. RH factor, right, because of pregnancy, right? Because the baby's red blood cells are different from your red blood cells. So hemolytic disease of the newborn, right, is this type of hypersensitivity. And again, it has to do with the antigens on the cell, on the surface of that red blood cell. And we say antigen, but what's another thing that you could say when I say antigen? What does that mean to your body when I say antigen? Something that's going to cause our body to generate an antibody. But why is that? It's foreign, right? Our body's not recognizing it as self, right? It's foreign. It's different. If it was the same, would we run into a problem? No, right? So if we match blood, right, if we give you what you already have, then are we going to have a problem? No, not unless you're experiencing autoimmune disease, right? You shouldn't have a problem because it's the same as you. The problem is everybody's blood the same? No, we found that out the hard way for some people, right? So for cytotoxic, it makes sense that the antigens are attached to the cells, right? Because we're going to destroy cells. Yeah.
So why is that if certain red blood cells have antibodies?
Yeah. In the interest of time, I'm not going to answer it today, but I am going to answer it Tuesday. I promise you that. I'll even do some Punnett squares if you want. Right? So, for the next one, antigen-antibody interactions, these are mostly going to be relatively soluble. For delayed cell-mediated, the antigens can be soluble or they can be cell-bound. So we could be talking about destroying cells, right, that are displaying foreign substances, or we can be attacking foreign substances that enter into the body, but with a cellular response, right? Because remember, when we talk about adaptive immune, you can produce antibodies, which is a humoral antibody response, Or you can do a cellular response, right? You can have cytotoxic T cells being involved as opposed to antibodies. Or you could have both, right? But in this case, with this response, it's cells, right? It's cytotoxic T cells. Okay. Remember I said 3 of these all involve antibodies. And in the first one, it tells you what antibody, right? It's IgE. Remember, Erica hates this one. Because this is the one, right, that this type of hypersensitivity we commonly refer to as an allergic response. For cytotoxic, how is it that we know that the red blood cell we just gave you from someone else or from your baby is different? What class of antibodies are binding to these red blood cells? IgG in the case of hemolytic disease of the newborn, and why is that? What's special about IgG? So I'm a mom, right? I'm pregnant. Why is it the baby has to worry about IgGs that I'm producing? Does my blood mix with my baby's blood? No. But do products from my blood go into her blood or his blood? Yes. Can IgG do that? Yes. IgG is the one class of antibody, and it's why we have hemolytic disease of the newborn, because it's the one class of antibody that can cross the placenta. You don't have to worry about matching your baby on the ABO blood group, because the antibody class you make against that is Ig. Which one does not leave the blood? IgM. So these guys, depending on what you're talking about, it's IgG and or IgM. Our main antibodies that are in our body. Why not IgA? That's in your secretions, right? That's not in your body, per se. What's the one that is everywhere in your body? In your fluids, in your body, in your blood, in your tissue fluids? What's your main protective antibody? Ig. No, inside your body. G. It's G. Are there any antibodies involved in delayed cell mediated? No. Hence the name. Cell mediated. What type of cells are responsible? So if we're dealing with antibodies, where did those come from? B cells. What's the answer for the next one? B cells. The next one?
B cells?
B cells, because we're talking about antibodies, right? If you've got production of antibodies, where did those come from? A B cell made them, right? That's the cell type that's responsible for this hypersensitivity. That B cell didn't make that antibody, we wouldn't have this problem, right? For the last one, T cells. What other cells might be involved, right? So for this one, the problem with IgEs is they bind to mast cells and basophils. And they're the ones that degranulate, release the substances that cause the inflammation response that we see with type 1 IgE mediated hypersensitivities, what we commonly refer to as allergy. For cytotoxic, think about this. If you have a cell with an antigen on its surface, right? And an antibody binds to it. Who's going to destroy that cell because an antibody is bound to it? Right, natural killer cells, right, are going to kill these cells. Then there's another system in our body that becomes activated, so I think we put it in the next, yeah, we put it on a mediator, so we'll get to that one. Immune complex mediated. Who's supposed to clear up these things when we've got a whole bunch of antigen and antibody complexed with each other? Who's going to gobble this up and eliminate it from the system? Macrophages. Right? So other white blood cells, but we could say, you know, macrophages are one of the players that should be cleaning this up. and again, other white blood cells will get online with T cells. So if T cells are killing in that area, sometimes they'll also trigger macrophages to head into that area and help them out. And especially for killing cells, right, if cytotoxic T cells are killing cells, the macrophages are going to clean up that mess, right? So they'll usually be called into the area. You wouldn't necessarily have a true inflammation response, but you have what's referred to as an influx response. So what mediators are involved? So most people know what's the major mediator that's released by basophils and mast cells that lead to the inflammatory response you see with an allergy? Good old histamine. There are other ones, right, but we're going to stick to that one. For cytotoxic, right, so we've got cells with IgG binding to them or IgM. What system in our blood that's inactive that can be activated via the classical pathway? Complement. And remember, what are the three main outcomes of complement activation? Opsonization with C, what? C3b is the opsonizer, right? And then we also get the production of C3a and C5a. What's their role? Inflammation. And then we can get a formation of a complex that we call what? It's going to poke donut-shaped holes in what part of a cell?
Cell membrane?
Membrane. So we call this complex membrane attack complex, MAC for short, right? So guess what happens to these red blood cells? Natural killer cells are going to kill them if they're coated in IgG. If they're coated in IgG or IgM, complement's going to get activated, and those cells are going to be lysed by the MAC complex.
For the cytotoxic, is it exclusive to red blood cells, or is it just any cell in the body?
Repeat.
For the cytotoxic sensitivity,
Right?
is it exclusive to red blood cells?
Yes. So this particular hypersensitivity is red blood cells. When we talk about transfusion reactions and hemolytic disease of the newborn. Not to say it can't happen probably with other cells, but it really causes a problem when it happens to red blood cells, right? When we're trying to do transfusions, or if you have the problem of your baby being different from you. Okay, so immune complex mediated, how do our immune cells communicate with each other? What's the general name we give to those molecules? Cytokines, right? But complement can also be activated for this one. So the next one is actually cytokines. I got ahead of myself. So the other thing is reaction time. So how long do you think, if we're saying immediate, what does immediate mean to you? Like seconds, right? Seconds to about 30 minutes. And I can tell you, this can happen in seconds. Because we'll talk about next time how you're primed and ready for this to happen. Cytotoxic, right? This one is going to be hours to days, as well as immune complex mediated. And then as delayed implied, right? So if we've been talking about seconds, we've been talking about hours, maybe days.
Months to years?
No, days. Still days, right? Still days, right? But multiple days. We're talking about 2 to 3 days. When we purposely put a foreign substance in your skin to perform the TB test, we're looking for this type of reaction. We're looking for a cellular response to those proteins associated. Your TB test is a delayed hypersensitivity reaction. If your immune system does what? If it recognizes it, right? And so if it's the first time you've ever been exposed, Are you going to have a reaction in 2 to 3 days? No, because you got to prime or sensitize, in this case, the system ahead of time. So people who experience allergies, do they sometimes have a reaction in their skin? Yeah, right? Especially if their skin is affected, right? If it comes in contact with what they're allergic to. With cytotoxic, remember, we're mainly talking about red blood cells. So you're going to see this in the skin? No. Immune complex mediated. So these are antigen antibodies. Is there potential to see this in the skin? Maybe. Depends if the complexes are happening in the skin. If they do, it's referred to as an arthrous reaction. And then, as I said, if it involves the skin, you can get a skin reaction. The TB test is an example of that, right? Where you'll get a reaction in the skin because we put the pathogen or the piece of it in your skin. This is an excellent table that summarizes some of the main components. And as we kind of talked about it, right, we're going to go back, and this should actually be reviewed for you guys on Tuesday, right? Because we're going to talk about, as we just did, how is it do you make an antibody, right? And what's the problem here? So with type 1, what's the problem? Because it's the particular antibody it is, which is IgE, and what it's responding to can't hurt you. But is this response what our immune system normally does? Yeah. Making antibodies against something foreign is very common. The problem is this particular class tends to be made against things we don't need to respond to. But it's the same progression, y'all. Antibodies are always made the same way. We're exposed to something, we make antibodies, right? So what happens when you're exposed again? Your memory cells make antibodies again, and again, and again, right? And that's the real problem for some of us, is this response keeps happening and gets more aggressive each time it happens. Something to think about. So we're going to go over these 4 hypersensitivities on Tuesday and talk about a couple of other topics, and then that's it. We're done.